In this last video of our macroevolution section, I want to take a moment to refresh your memory on paralogous genes, which we talked about during our microevolution video series. Remember that gene duplication can increase novelty or increase genetic diversity of that genome. Paralogous genes themselves, if you remember, are duplicated within a species lineage, creating new genes with new functions, but still related to that ancestral gene. Uh, One of those copies can serve the original function or purpose, while the other duplicate copies are quote unquote free to diverge in function. They can be mutated or modified and changed over time to add to the complexity of the organism's phenotype or diversity of its genotype. The example we looked at in our microevolution section were beta globin genes. These control the formation of the hemoglobin protein, which carries oxygen through our blood. Paralogous genes for beta globin, you can see that there are 5 here. There are 5 functional copies. They exist on human chromosome number 16. However, right in the middle of that series of 5 functional paralogous genes is a non-functioning copy of the beta globin. And this non-functioning copy of a paralogous gene is called a pseudogene. It's a section of chromosome that's an imperfect copy of the functional one, and it occurs in the same exact way as other paralogous genes through the accumulation of multiple mutations, but for some reason, one or more of those mutations created a non-functional version of this copied or duplicated gene. So in the middle of these 5 functional paralogous genes for beta globin, you have the beta globin pseudogene. Its DNA-based sequence is nearly identical to that of its functional neighbors, so it's easy to recognize in DNA analysis, but it contains a series of errors that keep it from working, keep it from functioning. These pseudogenes tend not to be expressed, that's the non-functional part, due to mutations, particularly in regulatory genes, the ones that control the turning on and off of genes. They are formed as a section of a chromosome that is an imperfect copy of the functional gene, as we just mentioned, but it typically doesn't happen from just one mutation. It has to happen through a series of mutations. It takes more than one. So in this illustration, you can see the original single gene that through the process of evolution over time, right, on the microevolutionary scale, we get paralogous genes, we get duplicated genes, one of which in future generations you can see maintains the original function of that gene, while other copies can diversify and be altered through mutation. In the case of a pseudogene, the set of mutations that occur within a copy make it non-functioning, basically impair it. They lack that regulatory gene, in this case, the green section, that would be able to turn the desired gene on and off. So it just doesn't work. So you may ask, well, why keep them around? Why isn't this thing selected against and snipped out of our genome? And the reason being, identical beta globin pseudogenes have actually been found in both humans and chimpanzees, leading to the use of them as essentially a fossil record within our DNA. So it shows us a common ancestry between more than one species, like in this case, humans and chimpanzees. Our genome was originally designed with 6 working copies of the beta globin gene. Therefore, the loss of one of them from molecular errors like mutations was really no big deal. All humans alive today are therefore descended from individuals in which those mistakes first cropped up. But oddly enough, what's really cool is that we're not the only organisms with a set of beta globin genes, and we're not the only ones with a pseudogene right in the middle of the set. Other primates like gorillas and chimpanzees have them too, and they're arranged in exactly the same way with exactly the same set of molecular errors or original mutations. So it's really cool that we can use pseudogenes almost like a genetic fossil record. In fact, pseudogenes are considered the quote unquote new fossils. Much of our genome consists of these fossil genes. They haven't gone away. So many have suggested that pseudogenes are molecular fossils that help illustrate and provide evidence of our evolutionary history. Included in this argument is that pseudogenes are genetic relics, right? They've lost their original protein coding function, which had been possessed by some ancestral creature. In support of this argument, evolutionary scientists point to the fact that pseudogenes are pretty common. They're scattered throughout the genome of all higher species of both animals and plants. And in particular, as mentioned in our last example, there are many similar pseudogenes found in all primates, us included, as well as our other great ape ancestors that we mentioned, like gorillas and chimpanzees. Another example I'd like to show you is that of the pseudogene for vitamin C. We and other primates possess this pseudogene. What does that mean? Well, normally in animals, there are three particular genes shown in this set, the last of which, abbreviated by GULO, G-U-L-O, stands for gluconolactone oxidase. This is the gene that actually helps our body synthesize or create vitamin C. We humans and almost all primate species are not able to synthesize our own vitamin C. This GULO gene is no longer functional, so it cannot create the enzyme to synthesize or make vitamin C, hence the red crossout. This is due to a mutation in the gene encoding for GULO, rendering that final phase of the protein creation obsolete. So ascorbic acid or vitamin C is inoperative. It just can't function. However, we still have the gene that codes for GULO, but it's a pseudogene. It's no longer activated. It just sits there. But this is another really great example of micro and macro evolution kind of coming into play. We have to eat fruits and vegetables high in vitamin C to get the necessary amount for normal healthy function, but that's not the case in all animals. You can see here, this is the genetic code of the working GULO gene in a rat. This particular mammal has perfectly functioning GULO gene and can create its own vitamin C necessary for a healthy body. So rats, unlike us, don't need to go eat fruits and vegetables to prevent scurvy or a lack of vitamin C. Their body naturally creates it for them. But in the four genetic codes listed below, humans, chimps, orangutans, and macaques, these are all primates. Notice this nucleotide deletion right here. This is amongst several other of alternate insertions of nucleotide bases. This is the collection, color-coded here, of all the mutations and changes to this GULO gene that make us humans, as well as other primate relatives, incapable of synthesizing vitamin C on our own. This is an example of a pseudogene. But we still have the entire set of the GULO gene, even though it's mutated. It shows that all of these mammals descended, the rat included, from a common mammalian ancestor, and that we share this common descent. So it can be pretty cool to use pseudogenes as a means of kind of genetic fossils. Continuing a discussion on pseudogenes, if we are able to reverse a point mutation that silenced that gene and turned it, that paralogous gene, and turned it into a pseudogene, we can essentially reawaken or reactivate that gene. When this occurs, atavistic or ancestral traits can appear. And atavism is the phenomenon in which a phenotypic trait, some physical structure, reappears in an organism after a period of absence. So it's not present in the immediate predecessors or parent or grandparent generations of that individual, but it did exist in ancient ancestors. And so it just kind of reappears. Um, this is any genetic trait that reoccurs after skipping several generations. So if you notice that, you or a sibling or a cousin has red hair or blue eyes, but your parents and aunts and uncles don't have it, don't have blue or red hair, but maybe a distant grandparent or great grandparent did, that's an example of an atavistic trait. It kind of skips generations and then reappears. When phenotypic traits are lost during the process of natural selection, the genetic information in the DNA that codes for them, again, is not lost. It's just inactivated through mutations of a pseudogene. But it means that other mutations in future generations could also act upon that pseudogene, essentially reactivating it, even after that gene or that characteristic lie dormant for so many generations. Another rare example, unique in humans, is people born with tail structures. This is extremely rare, but you know that you have a tailbone, you have a coccyx. And we, unlike our early primate ancestors, which had tails to help them balance on all fours and move through trees and things like that, we have lost it. It's called a vestigial trait. But while extremely rare, since 1884, human medical records have reported a total of 23 cases of human babies born with a tail-like structure, meaning that the tailbone does not terminate where it normally would at the base of the spine, but rather continues, and more vertebrae than normal extend from the base of that structure, creating a small tail. It can certainly be altered cosmetically. It can be cut off, and the person no longer has a tail. It is extremely rare, as mentioned, only 23 cases in nearly 200 years. But that's an example of an atavistic trait where some kind of port mutation was kind of reactivated from our ancient ancestor who had tails to modern day humans that have lost that phenotype, kind of buried in the pseudogenes of their genome, can sometimes get reactivated and reappear. Human tails are not the weirdest one, however. There are whales and dolphins that actually have hind limbs. As we saw in previous slides, whales descended from a four-legged terrestrial mammal. The hind legs became significantly reduced in size to the point where they disappeared externally, no external limbs, for better swimming, more hydrodynamic, streamlined body. But this is a dolphin that has been turned upside down, and you can see that this atavistic trait has reappeared. There are hind limbs on this dolphin descended from its extremely ancient ancestors, but somehow that pseudogene got flipped back on and reactivated. We can also see this in chicken that are born with teeth. You probably know birds don't have teeth. They have bills and beaks to crush their food, but birds did descend from ancient reptile-like ancestors, and reptiles definitely had teeth. So in some chicks, as you can see in this kind of embryo image here, this is the bill of an embryological chick that actually has teeth compared to a normal one that just has smooth edge to its bill. In addition to pseudogenes, we can also see that related organisms do share homologies. You should recognize this term. We talked about homologous traits before. Homologous traits are those that are inherited from a common ancestor. Not only can the physical anatomy reflect the relationships or kinships between organisms, but so too can their genes when shared from that same ancestral genome or genetic basis. One of those genetic markers that we can look for in related species is called a HOX gene, H-O-X. This is a type of homeobox gene. Homeobox genes direct formation of early embryonic structures. So when an organism is in the womb, developing from that zygote, and it's first starting to form a head, a neck, a tail, arms, legs, limbs, etc., The homeobox genes direct the formation of those physical body structures. Hox genes specifically direct the axis of the body, the head to tail axis. So if your head is supposed to form on top and not on the bottom of your body, then your Hox gene is the one that directs the head to be formed on top, and arms to be formed at the torso versus below the pelvis, that kind of thing. So it directs head to tail formations, ultimately informing body shape. And this has evolved in ancestors to all animals, with the exception of one. Cnidarians, as you learned about in lab, they're really unique, but still somewhat primitive or prehistoric creatures, less complex. They do not have Hox genes, which is kind of unique among animals. Hox genes are essentially considered the quote unquote animal gene, because we all have it, to almost all of us, with the exception of cnidarians, have it to direct our head to tail axis. So here you can see the color-coded chromosome of a fly. This is its larval stage, the embryonic fly, and this is the adult form. The color coding of the chromosomes along its Hox gene are also color coded in the body part formations from head to tail that you can see here, from the head, the little yellow spot, to the kind of torso, down into the base of the abdomen, kind of the tail portion here. And we see that too in mice, in another animal. So here you have the mouse embryo, again, color-coded by the sections of the Hox genes and where those structures are internally color-coded on the adult mouse. This is an example of a paralogous gene because the mouse actually has 4 copies of the Hox gene, all of which have been slightly modified to increase complexity of the mammal mouse versus the insect fruit fly. Continuing this idea that anatomy and genes reflect kinships, mutations on the same orthologous gene can cause similar defects in different animals. If you remember from our last lecture series, orthologous genes are those that diverged after a speciation event. So they're inherited in 2 different species from a common ancestor, but then after those species have diverged and have speciated, the ancestral gene can alter its function, even though it still remains somewhat similar to that ancestral gene. One example is the PAX6 gene. This is a gene that controls eye formation. During embryological development, the PAX6 protein that PAX6 gene codes for is thought to turn on or activate the genes involved in the formation of the eye itself, as well as the brain and the spinal cord. It's essential to the central nervous system. The PAX6 protein controls many aspects of eye development at the embryological stage. So after birth, the PAX6 protein most likely regulates the expression of various genes in the structure of the eye. So if there is a defect in the PAX6 gene, if there are mutations, in this case in the human, this is a normal PAX6 gene eyeball. This is a PAX6 mutated gene in a human. You can kind of see the clouded cataracts over this eye. But remember that genes can reflect kinships. We are not the only animal that has a PAX6 gene to control the formation of our eye. So along the top row, you can see normal eye formation, not just in humans, but in mice, type of fish called a zebrafish, and our Drosophila species of fruit flies. Here too, on the bottom row, you can see what the deformation of the eye structure looks like as a result of PAX6 gene mutation. So across species that at this level, at the species level, are completely unrelated. But as animals, they descend from a common ancestor that passed on this PAX6 orthologous gene. And so you continue to see mutations to this gene across species can have the same physical or phenotypic effects. The last example that we'll mention is a related set of fruit flies spread across the Hawaiian island chain. This is a great example of how neighbors, species that are physically living close to each other, can be genetically related. The distribution patterns of where these species live can reflect their patterns of evolutionary relationships. In this graphic, what you see down here are the main Hawaiian islands from east to west. The youngest islands are in the east. The older islands, due to plate tectonics, have kind of shifted and moved in the northwest direction. As new Hawaiian islands are formed in the east, species from the nearest older island, slightly to the west, are then able to colonize the new island and become reproductively isolated. That's what these gray arrows represent. So these three Drosophila fruit flies, these three species started out on the island of Oahu, but as Molokai and Maui formed as new islands, and this many millions of years ago, these flies from Oahu were able to move and colonize onto Molokai and Maui. And again, when the big island of Hawaii formed, they were able, through, as you can see, the gray arrows, move and colonize into this new patch of land. This conveyor belt kind of speciation process has allowed the Hawaiian members of fruit flies, this Drosophila genus, to radiate rapidly, forming multiple species that display extreme morphological and behavioral diversity across the islands. The phylogenetic tree that you see here to the left, this illustrates the inferred relations and relative speciation times in millions of years down at the bottom across the 7 island species. Fruit flies are not the only organisms that we see this pattern in. There is a video posted in your Canvas module about Galapagos finches. Definitely watch this video. It gives you a really good explanation of how patterns of distribution can reflect patterns of relations across Darwin's finches. And this won't be the last time that we see this example. We will find layers of evidence for evolution like this, kind of scattered throughout our remaining sections. Pay close attention to these patterns, especially in our lab units, as they will help explain the unique synapomorphies and diversification of the biodiversity of organisms that we'll cover.